Thank you very much, Saji. I'd like to thank the scientific committee for inviting me to give the talk. But I'm also in a scientific committee, so it's a bit self-serving. Yeah. No, but I wasn't. It was an independent selection, right? So really a pleasure to be here to share a little bit about the projects we do. I'm actually a microbiome and a parasitologist scientist, but one of my projects actually hinges or has some relevance to neurobiology, and perhaps that has some relevance to aging as well. So I thought I'll share with you some of the recent work that we're doing in relation to microbiome and behavior. And for many years since my undergraduate days, I worked on a mini project on this gut protist, and I used this as a model organism for my PhD studies. And when I started my career in the university, I worked on this for many years. But in recent years, it has, because it's a gut protist or gut parasite, we also asked the question, does it have any impact on the gut microbiota and the gut immune response? And indeed, it has. But today I'm going to talk a bit more on the serotonin pathway and how this organism induces some dysfunction and therefore some behavioral abnormalities. That's a brief background. It is a single-cell eukaryote. It's actually a very pretty parasite. This is a light microscopic image of thousands of these spherical unicellular protists in culture. Okay, it's very common in the intestinal tract, found in a wide variety of hosts, and it's estimated to colonize 1 billion humans worldwide. So it makes this the most prevalent protist of humans. And it's like most protists or parasites, they are, or the most, like most intestinal parasites, they are transmitted via the fecal-oral route, via contaminated food and water. Now, what caught my eye several years ago when my collaborator Andrew Roger from Dalhousie University, Canada, published this paper. He's a bioinformatics expert, and he studied the genome of Blastocystis. And to his surprise, in this publication, he reports that 1 in 40 genes of Blastocystis are acquired through horizontal gene transfer. So 1 in 40 genes do not belong to Blastocystis. They come from the human host. They come from bacteria. And they mainly drive metabolism and virulence in the protist. Now, as I looked through his list of HGTs or LGTs, I noticed an interesting enzyme, tryptophanase. And for those of you who study microbiology will know, tryptophanase genes are uniquely found in E. coli, and they drive the synthesis of indole from tryptophan, right? The manufacture of indole from tryptophan. And we noticed that Blastocystis harbors this particular gene. So we wanted to ask, does Blastocystis therefore also metabolize tryptophan to indole? And to note, tryptophan is an essential amino acid, so we cannot manufacture this in our gut. We acquire tryptophan mainly from diet, and to a small extent from some bifidobacteria that we find in the gut. To our surprise, when we expressed Blastocystis tryptophanase and we tried to do a tryptophan to indole reaction, we got hardly any data. We could not get any indole from tryptophan. At that point, my postdoc wanted to give up on the project. He said, it's a dysfunctional gene. But I recall that enzymes work bilaterally, right? They can work both ways. So I said, try the other way. And he said, oh, there's a lot of product. You can get a lot of tryptophan from indole. So in this assay, it's called the Michaelis-Menten assay. It's a standard kinetic assays for enzymes. You find that the Km value from tryptophan to indole is very high, which suggests a very inefficient reaction. Whereas the opposite reaction, when you load the reaction with indole and you look for tryptophan output, it's very low, which indicates high affinity between substrate and enzyme. So I had some use in the project. I didn't say, oh, you should give up because it's a problem enzyme. But beyond that, if it does produce tryptophan, which in itself is a very interesting finding, would it therefore have a role in serotonin pathway? Because the raw material, or basic building block of serotonin is tryptophan. Just to highlight that tryptophan is metabolized in the enterochromatin cells in the gut to serotonin by a distinct pathway, and this interacts with the vagus nerve. And this vagus nerve is actually your bidirectional nerve communication between gut and brain, and between the brain and the gut. We kind of had an idea that this would work because beyond the vagus nerve bidirectional signaling, blastocystis has also been associated with irritable bowel syndrome. And IBS is associated with dysfunctional serotonin levels. Beyond that, of course, serotonin is a neurotransmitter, and serotonin levels need to be homeostatic for us to maintain good sleep, good feelings of happiness and contentment. Okay, and also if there's any dysfunction, then there may be anxiety and depression. So there were many reasons to proceed with this project, and therefore I engaged a PhD student, Stephen Leonardi, to focus on this. But more than that, there was also some studies around the time that we were exploring this connection that showed that blastocystis is linked to increased anxiety in rodents and altered cognition in humans. But beyond that, there was no mechanism suggested or no basis for the association suggested. So we came up with these hypotheses. Can blastocystis meaningfully alter gastrointestinal tryptophan levels? Can this tryptophan levels translate to serotonin synthesis? Can this be replicated in the mouse model, therefore influencing features like behavior in mice? So I mentioned that we did this in a recombinant study, but does it occur at the cellular level? So do the organisms itself have this preferred kind of pathway leading to the increased production of tryptophan from indole? So we did a simple assay. We suspended the organism in media. We added either tryptophan or indole and looked for the cognate product by either calorimetric or fluorometric methods. So I'll just state that blastocystis can convert indole tryptophan at the cellular level, not just using the purified protein. So in this readout, if you add the organisms with tryptophan and you look for indole after overnight incubation, there is no increase. Using your model organism, E. coli, you can see the expected production of indole for tryptophan. If you do the flip experiment, if you co-incubate the organism with indole, you get a significantly more production of tryptophan, which, as I mentioned, is in itself a very unusual observation. And in E. coli, you don't get such a readout. So it sort of pans out quite well from, or validates the recombinant data. So just to say that blastocystis tryptophanase is adapted for the production of tryptophan. As I mentioned, other microbes can do this, but it's rare, and they don't use tryptophanase. So this is a very unusual tryptophanase. So the next question we ask, does indole and blastocystis, i.e., therefore the production of tryptophan, upregulate serotonin pathway genes or gene pathways in EC cell model? So I mentioned that enterochromatin cells are evolved to produce serotonin from tryptophan. There are in vitro models. The RIN14B cell is a cell line, is an established model. So again, the same thing, same setup. Expose this setup to RIN14B cells, and we do a gene signature readout for gene signatures of serotonin pathway. And again, just to say that the indolized blastocystis upregulates serotonin synthesis gene expression in RIN14B cells. So we're very pleased to see that this occurs. Now, I like to highlight that actually all you need to do is look for one gene. This is the key gene in the conversion of tryptophan to 5-hydroxytryptophan through tryptophan hydroxylase. This is a rate-determining gene and key to the production of serotonin. Now, in the control and in the indole-only condition, TPH1 is mildly increased, hardly anything. When we loaded it with tryptophan, we actually got a low reading, which can be explained by the fact that there's negative feedback. So I won't elaborate too much on this. What's important is if you add the indolized parasite supernatant, and obtain the supernatant, you get a significant increase in TPH1. If you sort of normalize it against the indole control, you get even clearer upregulation. And what's interesting beyond that is that the dopamine genes were also activated in this pathway. Okay. So does this translate, therefore, to serotonin synthesis? I looked at the pathway genes. How about the product? Again, similar setup, but we just identify serotonin via ELISA. So like a broken record, I'm going to say that indolized blastocystis does induce serotonin synthesis in Rin14B cells. If you do a readout for serotonin at basal levels, you do get some serotonin with just media, but with the control tryptophan, you get a significant amount. But importantly, with the indolized subtype 7b supernatant, serotonin is produced. To ensure specificity, we validated this by using 533401. This is an inhibitor specifically targeting TPH1, right? So this is a TPH1 inhibitor, and when you co-incubate the setup with this inhibitor, you abolish serotonin synthesis, even in the context of subtype 7b with indole and together with inhibitor. So it kind of validates the specificity of this output. So this is the highlight. I mean, so does it then therefore do anything in the mouse? So we collaborated with Zhang Yongliang. This is the same collaborator that Brian Kennedy works with. We colonized the mice with blastocystis 6 times over 2 weeks, let it recover, study behavior, and also obtain organs for tryptophan, indole, and serotonin analysis. So we did 3 behavioral tests. Tail suspension test is a test for depression. If you are depressed, you give up struggling shorter, right? Open field test is to see whether the blastocystis alters movement. Light dark box is for anxiety. If you're anxious, you spend less time in the light and more time in the dark, very much like humans. Okay? So again, around that time, a study came out that showed that there was some linkage between blastocystis and anxiety. If we look at the tissue level, I just want to go quickly through this. At colon and cecum, there was a change in the indole levels. They tend to go up. There was also increase in tryptophan levels in the colon and cecum of blastocystis colonized mice. In relation to serotonin, there were slightly different results. There was less in the cecum, but more serotonin in the colon. So we can't really say whether these changes we see are due to raised or decreased levels, but what we can say that compared to controls, there was a dysregulation of the serotonin pathway. And the key experiment that I wanted to show, which sort of led to this publication that I'm going to elaborate on later, is that with subtype 7 colonization of mice, there was a significant decrease in time spent in the light box for one of the subtypes, one of the strains of subtype 7. And this suggests some anxiety-like behavior. Transit wasn't affected. Distance traveled not affected, meaning that blastocystis did not impact motility, and the speed was not impacted. In terms of tail suspension tests, you might even say that they struggled a bit longer, right? But there was no significance broadly. When you look at the situation where there was a significance between light dark box and the serotonin levels, and you took the individual data points, whether it be in cecum or colon, when there's blastocystis and there is a higher amount of serotonin, there's less time spent in light box. So the correlation is quite tight between serotonin levels and anxiety in mice. So I have about 30 seconds left. I just want to say that we're quite excited to say that blastocystis can synthesize tryptophan, very unusual for a microbe. It can stimulate EC cells to synthesize serotonin. this results in perturbed behavior in mice. And of course, it shows that microbiota dark matter, of which blastocystis is a member of, i.e. the non-bacterial component, may exert abnormalities beyond their gut habitats via specific mechanisms. So this is just a summary of what I mentioned, and we just published this recently. This is the work championed by my PhD student, 4 years of labor, and of course, several papers leading up to this penultimate, or this ultimate paper. So this is the lab that I'm very happy to lead. Stephen is one of my latest students who graduated. I'd like to acknowledge former lab members, collaborators, and in particular, neurophenotyping core, who helped with the behavioral studies. Thank you very much.
Thank you, Kevin, for the nice talk. We have a lot of time for questions.
I made it in time, so I get a good score.
Yes. There is one question.
Yes,
yeah, please.
please. Yes. This is a very good question. So if I had my way, I would have done this in a germ-free mice, where I could just control the serotonin metabolism and the parasite itself. Now, you rightly pointed out that the microbiome was a normal microbiome, and we've also shown in previous studies that the microbiome is altered in these mice. So it is quite likely that blastocystis is part of a bigger effect on this serotonin pathway. But do note that in terms of producing tryptophan, very few organisms do it. So if the diet is the same and only blastocystis is added in, the only tryptophan producer beyond the diet is blastocystis, right? And if it's a high dose colonization, we have control for other features by virtue of the fact that this is a unique pathway.
Yeah. Another question is about the two strains. So actually, I found that actually the outcome for serotonin and indo, actually for these two strains actually are different. Correct me if I am wrong.
Yes. So we sort of had one strain, but I asked my student to check the other strains of the same genotype that has some heterogeneity in terms of tryptophan production. So we chose another strain with higher tryptophan levels. Okay, and this is the one, fortunately, was able to exert a phenotype. Yeah.
Thank you.
Thank you.
This is maybe off topic. One of the things linked to aging a lot is HRV, heart rate variability, which you can measure in mice and is highly linked to vagus nerve regulation. I wonder if it's interesting to look at that in this context.
Very interesting, yeah. I would like to take this further. This is just the start of a series of experiments. I just presented this in Spain at the microbiome conference. And one of the senior people in the audience, linked to your question, said, this increased anxiety, is it necessarily a bad thing, right? Because high serotonin is supposed to be good, right? So I said, yeah, maybe the mice are happy and anxious. I'm happy and anxious right now. I'm happy with you. I'm anxious to get this over with, right? So it is possible to have happy anxiety, right? So it's true, there are more things to explore, more things to consider, and we can take it up further as well. Yeah.
I have a comment. I think it depends on which neural network the serotonin is. For example, in case of amygdala, prefrontal cortical network, then you will be in trouble. Otherwise, it's okay.
Correct.
Okay, we still have time for questions? No? Okay, thank you, Kevin, for the
Thank
very
you
nice
very
talk.
much.